The title of the presentation is High-Density Peptide Microarrays, a Versatile Platform for Autoimmune Research. So again, please use the question pane to type your questions, and we will also share these slides and the webinar file after the presentation. Okay, Jill, I'm handing the cursor over to you. Please take it away.
Okay, great. Thanks so much, Karsten, for the introduction, and thanks everyone for joining. So for the agenda, we're just going to start off with a brief company introduction so you can get to know us a little bit better. And then we are going to go back to basics and talk a little bit about how PEPperPRINT epitope mapping works, including our linear conformational and substitution scans. And then, as indicated, for the focus of the webinar is on our autoimmune research portfolio, so we'll just let you know what arrays we have available to fit into your workflow. And we'll give just some real-life case studies in recent publications. And then, as Karsten mentioned, we'll have a short Q&A at the end, and the presentation itself is around 20-25 minutes. So the company is based in Heidelberg, Germany, which is about 1 hour south of Frankfurt. It's a really beautiful part of the country. I was visiting there just a few months ago. It's privately held, independent company. And the company was based on a proprietary laser-printed peptide microarray technology that was initially published in Science in 2007. So the founders incorporated and had our first product launch in 2011. And now, just 6 years later, we have satellite offices in Cambridge, Mass., San Diego, California, which is where I'm located, Brazil, and Japan. We offer over 30 standard microarrays, and a majority of our services come through customized microarrays, which I'll be talking a lot about later on. We also offer solubilized peptide pools, and the development of ELISA kits that may eventually be used in a diagnostic setting. So how does this peptide synthesis on a chip work? Well, it starts with the development of a peptide laser printer, which is seen here in the upper right corner. It's around 4 to 5 times the size of an office laser printer. But instead of having 4 color cartridges, which are present in an office laser printer, our printer contains 24 amino acid cartridges, which are seen here in the bottom right. So 20 for the natural amino acids and 4 for any unnatural amino acids. So each cartridge contains microparticles with 1 activated amino acid embedded in the toner material. So after simultaneously printing of all the amino acids on the glass slides, all the microparticles are melted, which is seen here on the upper left side, where the particles are printed. This is the glass slide here, melted and coupled, washed, deprotected, and this process is repeated over and over until you have your peptide of interest printed on the slide. So the benefits of this technique are its unique digital flexibility in terms of peptide content, high spot density, as well as significantly reduced material consumption. So the process of this direct synthesis of peptides on the chip is really unique to the field of peptide arrays, and really allows a competitive advantage that saves material, time, and costs. So a normal standard peptide array requires pre-synthesis of peptides and then spotting those peptides on the chip. Where as I mentioned, we have direct synthesis of peptides, and so we skip that middle step. So this increases the amount of time it takes to make the chip, increases costs. Well, in the standard arrays, it increases the cost because you pay per peptide, and increases the waste and decreases the amount of peptides per chip, resulting in a low resolution mapping. In essence, what the PEPperPRINT technology allows is to think of it as around 10 of thousands of peptide ELISAs in each microarray. So the PEPperCHP peptide microarrays can be used for various applications, as indicated here, including vaccine development, which, as Karsten mentioned, I used while I was a postdoc at Scripps, biomarker discovery, infectious disease, cancer, and clearly autoimmune research. But I am going to just take a couple slides just talking about kind of the bread and butter application, which is antibody analysis, and this can be used which is applicable for each of these all individual applications. Okay, so we offer linear epitope mapping and conformational, which for both we have maximum peptide-peptide overlap, which results in high resolution. And then our PEPperMAP epitope substitution scan, which allows for an in-depth analysis of the epitopes and identification of essential and variable amino acids. So for the next couple slides, I'm just going to be talking about each of these arrays. Okay, so this is a schematic of how epitope mapping works. So it's a little bit busy, so just bear with me because I'll work us through it. So on the top here, you have, you start with your amino acid sequence of the antigen to which antibodies bind, or the sera. That sequence is then translated into overlapping peptides. And we routinely synthesize 15 more peptides with a peptide-peptide shift of one amino acid, which equals a maximum peptide-peptide overlap of 14 amino acids. As mentioned, the peptides are directly synthesized onto the glass slides. The array is subsequently stained with the target antibody or challenged with the sera of interest, and then you add a secondary antibody with a fluorophore on it so you can identify your epitope. This is a schematic of what it may look on the scan. The image of the array is taken with a microarray scanner, and since we are using spot duplicates, each signal appears twice. After the data acquisition using a specialized software, it immediately calculates the common motif in the peptide hits, which results in the epitope of the target antibody. So what you see here on the left is an example of a monoclonal stain, which it identifies a single epitope, and then on the bottom is a polyclonal sample recognizing multiple epitopes. And they're both flagged with control peptides visible as red and green ribbon. Okay, so here's an example in the context of comparing the PEPperPRINT array to a standard array. So we performed two arrays with the antigen CMPA that was translated into a 15 amino acid with maximum peptide overlap of 14 amino acids, which is the PEPperPRINT on the left, and then the standard with the 15 amino acid with an overlap of 11 amino acid. They were both assayed with human serum of a sclerosis patient followed by staining with a secondary antibody. And as you can see, the serum showed a strong response with excellent signal-to-noise ratio against the epitopes in the antigen CMPA with the PEPperPRINT. Two distinct epitopes could be clearly identified using the maximum peptide overlap, where you really weren't able to see that in excuse me, the standard, where you were only able to identify a short 3 amino acid PTP epitope. So our data show that the maximum peptide overlap is required for unambiguous identification of two neighboring epitopes and demonstrate the advantages of PEPperPRINT's high-resolution epitope mapping compared to our competitors. So our another benefit is our technology is really flexible. We can perform the assays with monoclonal, polyclonal antibodies, synthetic or purified antibodies, or hybridoma supernatant, plasma or serum, used with any species, just the secondary antibody with the fluorescent tag needs to be available, and also different isotypes. Okay, so this, as anyone who is familiar with antibodies, worked with antibodies, understands that many epitopes rely on protein folding and form so-called conformational epitopes. So now I just want to talk a little bit about our conformational epitope mapping. So because a lot of these conformational epitopes can hardly be detected with a standard microarray based on linear peptides. So a common strategy to mimic conformational motifs is through the introduction of structural constraints by peptide cyclization. So, for example, as shown here is a schematic of CD20. This receptor is expressed highly on B cells and B cell tumors, specifically non-Hodgkin's lymphoma. It comprises of 4 transmembrane domains and 2 extracellular domains. One extracellular domain is a very small loop of 8 amino acids here. And the larger loop is structured by a disulfide bridge and comprises roughly 40 amino acids. So these epitopes can't be detected based on linear peptides, but conformational peptides. So what we do is print the peptide as briefly described in the previous slide as a linear peptide, and then add that extra step of cyclization. So for a proof of concept study to show that this actually works, we chose the interaction between rituximab and a therapeutic monoclonal antibody that's directed against extracellular domain of CD20. So in 2007, JPC published the crystal structure of rituximab and CD20, and they found that the rituximab binds to CD20 somewhere in this 15 amino acid loop, but wasn't specifically sure what the epitope was. So to get more of an in-depth view of the core motif, we employed this conformational epitope mapping. So we started with the microarrays. We also did a linear array and then challenged the linear array of that fifth amino acid loop and challenged it with the rituximab. And as you can see here, there were no hits, which was to be expected. Then on the bottom, this is showing the array of where the peptides were actually cyclized at 7, 10, and 13 amino acids. And then they were challenged with rituximab, and you can see there are clearly defined hits. So the poor reaction of the cyclic 7 amino acid could be explained by the actual length of the amino acid epitope, which we know is 8 amino acids, and the increase in signal intensity from the 10 to 13 amino acid loops likely results in better mimicry of the native 15 amino acid loop in the CD20. Okay, so now moving on to the substitution scan. So we're just going to use the same case study, the rituximab CD20, to illustrate how this works. Okay, so what we did is to take the So we took that part of the extracellular domain of CD20 and we printed onto the microarray, and we performed the epitope substitution scan. So this is in contrast to a typical alanine scan in which you substitute each amino acid in your peptide of interest with an alanine and see if it binds or if it doesn't bind. So this, we actually exchange each amino acid with each of the 20 amino acid, which dramatically increases your data analysis and gets a much more specific understanding of essential and non-essential amino acids. So here we have the part of the extracellular domain of CD20, and you can see in the N-terminus that there's a lot of tolerance for amino acid exchange. But here in the actual epitope, clearly there isn't so much tolerance, but interestingly, we were able to identify the essential amino acids of N, P, and S, which didn't tolerate any amino acids. So the kind of exciting or important part of this assay is that this can be used for understanding for patient stratification assays in which a patient has B-cell lymphoma and is considered a candidate for a rituximab, but they want to check to make sure that they don't have a mutation in this part of the antibody, because if they did, then they would be excluded from that therapy because it simply would not work. Okay, so those are our 3 assays, and I want to talk a little bit about specifically of our autoimmune research portfolio. So we have a number of standard arrays which are pre-printed, and they're all the peptides are based on the immune epitope database from the La Jolla Institute of Allergy and Immunology, which is based here in San Diego. And it's a free resource that's NIH funded. So we have the Autoimmune Epitope Microarray, the CCP Microarray, our Human Epitope Arrays, and our PEPperCHP Sera Arrays. In the next couple slides, I'll talk specifically about each one. And then as mentioned previously, we do have customized arrays which are flexible and made to order, where essentially whenever you have whatever peptide or antibody you have of interest, we just work with you to make a specific array on your needs. So this is a case study using the Autoimmune Epitope microarray. So this contains epitopes that correlate with a variety of autoimmune diseases, including Crohn's, diabetes, rheumatoid arthritis, Sjogren's syndrome, lupus, and many more. So in this example, the serum of a 56-year-old female diabetes type 1 patient was analyzed with this array. And you can see with the IgG in red and the IgM in green, the antibodies were profiled simultaneously on the same chip. So the patient showed high IgG titers against commonly found Epstein-Barr virus and polio antigens, as well as against the protein tyrosine phosphatase, IA2, which is a known type 1 diabetes autoantigen. Also, a moderate IgG response against calrectalin and various IgM responses against Sjogren's syndrome epitopes were observed. So this is really exciting in that it was opening up a new putative diagnostic marker for type 1 diabetes. So the IgM antibody profile of the serum corresponded to less mature immune responses, as expected. Although the general intensity level was slightly lower, the response pattern was more complex, possibly due to cross-reactivity of IgM antibodies. As with the IgG profile, we also observed some clear IgM responses against the epitopes of EBV. So in general, with higher sample numbers and particularly a comparison of patient and control groups, this PEPperPRINT autoimmune epitope array could be used to identify different serum biomarkers for autoimmune patient groups or discover new autoantibody profiles that correlate to a specific disease type. So this is our cyclic citrullonated peptide microarray. So this is useful for studies in rheumatoid arthritis. I'm sure most of you know that rheumatoid arthritis is one of the most frequent autoimmune disorders, and significant progress of serological RA diagnostic is based on the detection of CCP antigens. But as you know, it's the response of RA patients is very diverse. So making the diagnostics quite difficult. But we have the PEPperCHP cyclic citrullonated peptide array, which has 337 different cyclic citrullonated peptides, including the corresponding arginine controls for a high-throughput screening of antibodies to these antigens in the patient's sera. So in this example, IgG and IgM responses against the CCPs in the sera of 4 patients in the healthy control were analyzed. So while no signals were detected in the healthy control, the RA patients showed common but also differential responses of known RA citrullonated proteins. So you could also, for the any sort of autoimmune response, use our human epitope microarray. So this array has around 30,000 peptides printed, again, all available from the Immune Epitope database, and allows for epitope-wide screening. So also isotype specific, comparing the IgG with the IgM. Here we had sera of a patient with lupus, and what we observe is another number of hits to viral antigens in Sjogren's syndrome. And finally, we have our PEPperCHP sera RA library. So in an effort to develop companion and IVD diagnostic tests, we developed this sera RA library, which contains over 100,000 linear and constrained peptides of known autoantigens, pathogen-derived so-called autoantigens, and a number of bacterial antigens. Importantly, it not only covers the natural peptides, but includes homocitrulline peptides as well, which is kind of a hot topic these days in RA. So this is the most comprehensive peptide arrays identifying RA antigens, including this homocitrulline, with unlimited flexibility. So we can easily add a number of new antigens. This is, so we have this made, but certainly if there's something specific you would want on this, this assay, this is something we can work through. So it's highly specific to your needs. So this sort of assay we would definitely need to talk to you a little bit more about it. Then finally, for our customized array, this, as mentioned, is probably 80% of the projects that we work on, is that since everyone has different questions they want to ask in their experimental design and different peptides and antibodies, we can simply talk with you to find out your specific needs and simply make the chip. And it's a very quick turnaround time. So in this case study, we tested 3 patients' sera for a sclerosis study versus 1 control sera. And what we found was a number of unique features that we have in the same in each patient's sera with the highest signal-to-noise ratio. So if you zoom in into this region, you can see that it's not only a single peak, but due to the high resolution, we can see that the peak is based on 2 different epitopes. So we also have, I wanted to note that we have an application note that's available on our website that discusses this project more in detail. I just wanted to mention this paper that was published using the Peptide Microarray, and I thought it is quite interesting. They were using the arrays to look to find new indicators for multiple sclerosis. But the interesting thing is that they not only used epitopes of human and viral antigens, but they also used random peptides simply to screen for signatures in patient serum in different sample groups, and they found some really exciting results with that. We are always coming out with new products. We do have a new Lupus microarray. I'm not even quite sure if it's on the website yet, but it is available, so we wanted to let you guys know. It has a number of lupus antigens and lupus epitope, again, based on the immune epitope database. It has, I believe it's over 5,000 peptides. So if you'd like to know more about that, we have this available. Yes, so in summary, why work with PEPperPRINT? So we kind of went out and asked some of our customers specifically what were, kind of your top 3 reasons why you chose us. So the general consensus is first, customization. We really have the technology is flexible, but the company itself is really, we're founded by scientists and we hire scientists. We understand everyone has different needs and we really want to compromise and help you advance your research. Quality, we have, I hope it came across, but the peptide arrays with the technology of direct printing onto slides results in excellent signal-to-noise ratio, which outperforms competitors. You can really pinpoint specific epitopes of your antibody or sera interest. We are ISO 9001 certified laboratory, and also the service. We have very fast times. We have thorough scientific support. Not only, out here, it's very multi-layered. Out in the field, sales representatives like myself and Karsten, but also internally, we have a service team. And also on some of the more trickier projects, which I've had a few, the lead R&D actually came on the phone and talked with the customer directly to find out specifically what we needed to do to get their assay to work, which was quite impressive. So for more information, please turn to our website. We have a lot of tutorial videos and webinars, and the application notes, I mentioned a few. We are in social media, and of course, you're more than welcome to reach out to any your representative, sales representative that's on the contact page on the website, or internally to headquarters. Kind of depends where you live. And then with that, we're all done. I'd just like to thank you for your attention and ask if there's any questions.